Welcome to Brain Ponderings. I'm your host, Mark Matson. Today it's my pleasure to introduce you to a kind of a pioneer and major player in the cancer field, Professor Ron DePinho. He's a professor in Department of Cancer Biology at the University of Texas MD Anderson Cancer Center in Houston. He holds an endowed chair there. He was actually president of MD Anderson, which is a very widely known, prominent cancer center in the country. He's also launched this cancer moonshot program a while ago, which is a major effort. Some of you may be asking, why do I have Ron on the Brain Ponderings podcast when he could be studying brain cancer, I guess, which he has some, but has strong interest in aging. He'll talk about that, how he got interested in first cancer, then aging, and then the brain and brain aging and neurodegenerative disorders, which we'll try to focus the conversation, most of it, on that. Welcome, Ron.
Well, thank you very much, Mark, for having me.
You were a long time in New York. I understand you were born and raised in the Bronx, and then most of your college, undergraduate, then you went to medical school, did residencies, and then postdoc research, where I assume you really did a lot of bench work there. And so talk about kind of how it evolved that you stayed, and then you went to Texas from there, is that right?
I went to Boston.
Oh, you went to Boston?
Yeah,
Dana-Farber,
Dana-Farber, and Harvard Medical School.
Okay. So were either of your parents physicians or scientists?
No, my parents were, Portuguese immigrants. They came to this country for a better life. They grew up poor, uneducated. They came here, had a very strong work ethic, taught us core values, the value of education. And so it's the American dream, right? Of having individuals come, and my dad ended up starting a construction company and real estate, so on and so forth, but really was focused on helping others, and in particular his family, to achieve a better life than they had back in the old country.
Yeah, that's a fantastic story. Parents are so important, and if they emphasize reading, getting you to read and get a lot of knowledge in your head when you're young, and then with encouragement, that's all you need, really.
Exactly.
And so when did you first get interested in cancer?
Well, I started my postdoctoral research in Fred Alt's lab, who was one of the great molecular biologists at the time and still is. And we were cloning new oncogenes, MYC family oncogenes like NMYC and LMYC. And one of the things that was known at the time was that these oncogenes really dysregulated cell cycle. And there was just one hallmark of cancer back then. One cell became two. Things like angiogenesis, apoptosis, and so on were not really on the radar at that time. And one of the things that I noticed was that these oncogenes were expressed at different times of development that didn't really correlate with proliferation. They're expressed in different cells at different mental stages. And I thought, well, maybe these oncogenes do more than just regulate cell proliferation. Maybe they regulate developmental decisions. We now know that oncogenes regulate stemness and things of that nature, important hallmark of cancer. So I really was very interested in developmental, what I would call oncobiology. And I realized at that time that the only way that I could properly study that was to exploit the experimental merits of the mouse. Back then, transgenics were just being formed. Knockouts were not yet possible. But I felt that that was going to be a platform that would allow me to dissect these complex developmental genetic events that occur at the organismal level. Being a physician by background, I was more interested in organismal biology.
Sure.
Back then, at the era of the dawn of molecular biology, it was all about reductionism, single genes, a lot of biochemistry, and an in vivo experiment was HeLa cells. So this was so I wanted to pursue this angle. Actually, everybody advised me not to do that, that my career would be over because I would be spending all of my time just engineering mice and I wouldn't be competitive. But I really wanted to answer the question. And if I was going to fail answering that question, that was okay with me.
Okay, and so what were some of your first major findings in mice then?
Yeah, so basically, among the things that we looked at and that we increasingly got more and more involved in cancer specifically was that I'll give you a nice story, which was Tyler Jacks was presenting his RB knockout at a conference.
So this is
And
retinoblastoma.
exactly, and it's a very important cell cycle regulator. And in the RB knockout, I noticed that from his slides that the mice had nuclear cataracts. And I knew immediately that that was a sign that differentiation was affected. And so I collaborated with Tyler and we noticed.
And are those, so nuclear, it's also a major problem with aging.
It is.
Yeah.
Nuclear cataracts in particular are more congenital for the most part.
Oh, I see.
Essentially, the middle part of the lens, which is post-mitotic, in the RB knockouts had proliferative cells. And so that was one thing, is it disrupted differentiation and it maintained inappropriate mitotic cells in the lens.
Oh, I see.
And we love the lens because the lens has regional compartmentalization of proliferation, differentiation, and terminal differentiation. So in the center of the lens, they were still undergoing proliferation and they were not differentiating into elongated lens fiber cells. And
Well,
so
that's very similar in the general sense to the cerebral cortex.
exactly. And that's a great, this kind of regional compartmentalization. And what we found was, and this was the early days when we were just developing tunnel assay for looking at cell death, apoptosis, is that we noticed that not only were the cells proliferating, but they were undergoing apoptosis. And so we realized that cells, an important checkpoint for cancer is that as cells go into the cell cycle, there are mechanisms that cause cell death,
right,
that will eliminate those would-be cancer cells. And so we realized that perhaps what we could do, and this was the very first double tumor suppressor knockout ever, is that if we crossed the RB with P53, the two most commonly mutated pathways in cancer, that perhaps P53's function might be to allow those inappropriately cycling cells to survive and move towards becoming malignant. And so this was the first evidence that P53 functions as a checkpoint for inappropriately cycling cells and executes an apoptotic response. And that's essentially a checkpoint, another hurdle that aspiring cancer cells have to go through. So you knock out RB, you enter cell cycle, but you die. And then the loss of P53 allows this promotion of continued proliferation and so on. And we now know that virtually all cancers have RB and P53 knocked out as almost a rite of passage for the development of cancer. Of course, you need other things, and that's how we got into the next stage, which relates to the main focus here today, which is telomerase and so on.
And go ahead and, this is a huge area of research, telomerase, with a long history, but can you kind of go through the history of its discovery and its links to aging and cancer and so on? Because listeners and viewers have to have some background before we get into the brain, the
Sure.
nervous system.
So let me give you the historical context. So Leonard Hayflick put normal cells into culture. It was known that cells like HeLa cells and so on were able to go into culture and divide indefinitely. However, he showed that normal cells had a finite replicative potential, later to become known as the Hayflick limit. When Carol Greider and Liz Blackburn discovered telomerase and Liz telomere sequences, they were able to show that in the context of proliferating cells, normal cells, that where there is insufficient levels of telomerase to maintain and synthesize your telomeres, that there is a erosion of telomeres over time. That is, you can't fully synthesize the ends of your chromosomes when cells divide in the absence of telomerase. So now you kind of walk this telomere plank, and eventually that triggers the Hayflick limit. So that was Cal Harley and Liz Blackburn in a seminal paper that they published in 1990. And then Jerry Shea did a really critical experiment where, upon cloning of the human reverse transcriptase, they were able to show that constitutive expression of human reverse transcriptase was able to maintain telomere lengths and allow normal cells to go way past, indefinitely divide past the Hayflick limit. So this provided definitive evidence that the finite replicative potential of cells was governed by telomere dynamics and the presence or absence of telomerase. And that's one of the reasons why cancer cells are able to divide, is because they have reactivation and high levels of telomerase reverse transcriptase. And so, when we had done the experiments that I mentioned earlier with RB and P53, we realized that for cells to divide indefinitely, now that they can survive, perhaps the next checkpoint that they might incur would be the Hayflick limit. And at that time, I got a call from Carol Greider asking if we'd be interested in knocking out telomerase. They had just cloned the RNA component, which is the template.
Right.
So you have two key components: the telomerase reverse transcriptase, or the catalytic component, and the RNA component, which serves as the template for the de novo addition of telomere repeats. And so she had cloned the RNA component. We later went on to clone the mouse TERT gene that allowed us to knock out both components. Because what we wanted to do is to ask the question, we had all the cell culture-based work, but we wanted to ask the question, what is it that telomerase and telomeres do at the organismal level?
Yeah.
And at that time, mice, had very long telomeres, and humans had shorter telomeres. So it wasn't really clear that the mouse would be a good experimental system in which to really dissect the functions of telomerase and telomeres. And so we knocked it out, and the we in this case was Hanun Lee in my lab and Maria Blasco in Carol's lab. And they collaborated on the first knockout to show that actually you can survive without telomerase activity. That is, when you knock out the RNA component, the animals still live. And that was the first, part that you could survive without active telomerase. However, the mice had very long telomeres. So we suspected that we would have to take the knockout and cross it through successive generations of telomerase deficiency to, as their germ cells go through one generation to the next, that we would have a decline in germ cells. Decline in telomeres through the germline lineage, and eventually you'd have animals that would have more human-like telomere lengths. And what we found was that by the third generation, we began to see evidence of loss of telomere function. So we saw loss of the repeats, and coincident with loss of the repeats, we saw the fusion of ends of chromosomes. And that was something that McClintock predicted and Herman Mueller predicted in the 1930s. that telomeres are the caps of chromosomes and they help maintain chromosomal integrity. And even though they're free ends, they're protected from being free ends through proteins that cloak this and sufficient levels of telomere repeats to maintain this inert structure and repel DNA repair machinery so that they can't fuse. And so we're able to prove that. But the most interesting thing was what started happening on the organismal level. We began to see that coincident with loss of telomere function, that the animals began to experience organ atrophy, alopecia, impaired wound healing, impaired ability of the gut to regenerate, impaired immune system. And we also began to see neurodegenerative conditions. And that was the first evidence that DNA damage signaling, which is what telomeres are actually an instigator of neurodegeneration, something that Li Wei Tai recently reported in her analyses at the transcriptomic level. And Bruce Yanker went on to show that there are DNA damage foci in Alzheimer's patients and so on, some years after that.
I had Li Wei on one of my podcasts, but I was talking about gamma frequency activity in the brain, and
she is one of the true creative engines of the field. I've tremendous admiration for her.
Now, Ron, so what you've talked about so far is in proliferative cells, that the ends of the chromosomes with successive cell divisions, they get shorter, and then they reach a point where it's essentially like a DNA damage response. So neurons are postmitotic. So presumably, they're not dividing. So are there telomeres shortening during aging? And I would assume you can still get a DNA damage response at the telomeres.
Yeah, so that's a very important question. And again, this was influenced a lot by the cell culture-based studies. And indeed, that's a major reason why telomeres become dysfunctional, because they become ever so shorter, and then they reach a critical length where they can no longer get rid of, the DNA repair, processes, etc. However, it's important to keep in mind that telomeres are very G-rich. And Ross likes to modify guanines.
Yeah.
And you have enough G modification that you begin to what's called uncap the telomeres. Even though the telomeres might be a sufficient length, telomeres have this very interesting structure where they form T-loops, where the free end invades and forms a triplex. And that's cloaked by all of these proteins known as the sheltering complex. Well, if you start to modify enough Gs, then that whole complex three-dimensional architecture gets disrupted, and you can then lead to that. So you could have ROS as well, and so on. And those are all opportunities for cells to experience telomere dysfunction. And you made a very important point, which is it's a DNA damage signal. So when you uncap the telomeres, it gets recognized as a free end, and it sends off a DNA damage signal. Just like I mentioned earlier, where inappropriate cell cycle entry can activate p53-dependent checkpoint apoptotic responses, it turns out that p53, from the work of Mike Caston, when you have DNA damage, you can activate an apoptotic response as well.
Yeah.
And so p53 can sense many different signals: inappropriate cell cycle, DNA damage, and then p53 becomes activated. And in fact, we did an experiment where we crossed the p53 knockout to the telomerase knockout. And what we found was that we dramatically rescued a lot of the degenerative phenotypes of the animals. So now the animals cells were no longer undergoing apoptosis, and organs were no longer atrophic. And what we found instead was a rescue. However, now that the cells were still dividing and the genomes were very unstable, that then led to an increase in cancer. So it was the telomere dysfunction, the fusions, breakage events that occur at cell division when you have these fused chromosomes, that creates double-strand breaks, but the cells can survive because they have p53 deficiency. And you can cause amplifications, deletions randomly that might hit amplified oncogenes, deleted tumor suppressors, and eventually in this population of cells, you select for the right constellation of amplified oncogenes, deleted tumor suppressors to undergo that benign to malignant transition. And so another key hurdle of aspiring cancer cells, loss of RB for cell cycle dysregulation, and then deactivation of P3 to handle that cell cycle issue, but also enable telomere dysfunction to become a mutator mechanism for cells. And in fact, mice do not develop epithelial cancers. They develop primarily mesenchymal and hematopoietic malignancies, whereas humans predominantly develop epithelial cancers. Well, why? Humans have shorter telomeres, whereas mice have long telomeres and more promiscuous expression of telomerase. And as a result, that is an effective mutator mechanism in humans, but not so well exploited in the mouse. So they can't develop the double-strand break-inducing condition that generates the large number of changes needed for epithelial cells to become a cancer cell. And so that was the third major contribution that we made to the cancer field, but also it set us on a path to study the degenerative side of the equation. That is, if you think about the core pathway of aging, you can either get dysregulated and get immortality, i.e. cancer, or senescence and degeneration leading to degenerative conditions and aging, right? Yeah. And so virtually everything that we know about aging itself modulates signaling flux through a pathway that connects genotoxic stress like telomere dysfunction, the ability to make mitochondria, that is, mitochondrial dysfunction is an important aspect of aging, neurodegeneration, and so on. But also mitochondria play very important roles in ROS regulation. Yeah. But also this pathway regulates oxidative defense genes. So when you dysregulate this pathway in one direction, you can get immortality, knock out p53 or deactivate p53 with sirtuins, for example, or you can get a degenerative condition. And so that's something that we were involved in discovering is this core pathway.
So you mentioned ROS, reactive oxygen species, oxygen-free radicals, and one of the early theories of aging, Harman theory, was there's this accumulation of oxidative damage to the molecular components of cells, be it proteins, DNA, lipids. But cells have really effective antioxidant defenses. So neurons are they experience a lot of oxygen-free radical production, just like muscle cells when you exercise them. Right now, Ron, you and I are generating a lot of reactive oxygen species in neural networks in our hippocampus and cerebral cortex. But one interesting thing there that we've done a lot of work on is, so exercise, for example, exercise is really good for your health, but during the exercise, it's a stress. It's actually a huge stress on the cells. But if they don't get that stress, in the long run, it's a good stress. They don't fare as well because they're not upregulating their intrinsic defenses against this. Have there been studies looking at how exercise, caloric restriction, fasting, things that tend to help you age better, how they affect I mentioned they bolster antioxidant defenses, but do they impinge upon telomere-related pathways, p53-related pathways?
Well, there's a lot to unpack there.
Yeah, I know.
Let me distill it down and give you, and I know you know this, but so let's just look at this axis that I briefly mentioned, because everything you mentioned really impinges upon this core axis that we had discovered about 15 years ago. So there, you could start the aging problem cascade of events that leads to degeneration by interdicting almost any component of this pathway, that once you reach a critical threshold of dysregulation in any component of the pathway, you create this feed-forward loop that leads to significant pathological degeneration. So what happens, and if you just first of all look at the pathway, so let's just say that you have accumulating genotoxic stress at telomeres, and you're modifying your Gs. You might be eroding your telomeres, but you're also modifying those Gs. So over time, those do not get repaired. You reach a critical threshold in cells that then instigates a dysfunctional telomere state. That then tonically signals to P53. And now P3 is tonically signaling and tonically activated. When P53 is tonically activated, it can do a number of things, but one of the key things that it does is it represses PGC alpha, beta, 1 beta, and 1 alpha and 1 beta. Mm-hmm. PGCs are the master regulators of about 20 genes that regulate oxidative defense, and regulates almost all genes that make mitochondria and make mitochondrial work. Yeah. So let's just imagine that you've accumulated DNA damage sufficient amount. Now you're tonically signaling P3. Now you're reducing the amount of PGC. What does that do? It reduces the number of mitochondria that are formed, but it also makes the mitochondria that are there not work as well. In addition, you now have lower levels of oxidative defense. Now, those two things are very, very problematic because mitochondria act as a sink for free radicals, converting it to CO2 and water. Yeah. So if you now have a reduced pool of functional mitochondria, you have more ROS, but less oxidative defense. What happens now is you injure the telomeres even more. And you now signal to P3 even more. You repress PGC even more, and you cause mitochondrial dysfunction even more.
And so you have reduced ATP supply, which
exactly frailty, weakness, sarcopenia, and so on. Correct. And also diminished neuronal function, as an example. And so virtually all cells, your diminished cardiac function, so on and so forth. And so that pathway. So you could either start at the level of telomere biology and say that's an instigator, or you could start at the level of impaired mitochondrial function, declining function over time, and there are many instigators of that, and that could start the cascade of events. Yeah. And I agree with you that we have a lot of redundancy in the oxidative defense system. And so that is likely not to be a rate limiting factor. But again, once that cascade gets going, you can diminish your defense systems as well. Now, that's a very simplistic picture that I just presented. And P53 can also regulate P21 and cause senescence. Senescence can cause local inflammation, and we'll get into this later. And that can cause tissue damage and tissue decline and diminished tissue function as well. You can get immunosenescence, so on and so forth. And so that pathway, you mentioned diet, exercise, and so on, many things that could be modified. What all of those things do, and this is concerned from C. elegans to humans, is this pathway of P53 PGC, everything regulates signaling flux through those components. So sirtuins, which are histone deacetylases, will deacetylate certain residues on P53 and PGC to cause P3 become more or less active, PGC become more or less active, and exercise. Same thing. It regulates those two components. It modifies those two components to regulate signaling flux through those components. And we not only show this biochemically, but we show this genetically. That is, you can do epistasis experiments to show that A is connected to B, is connected to C, and that requires the phenotype, the degenerative phenotype that you see. And so this all links up nicely. It's a little bit simplistic. And then we wrote a review in Cell in 2021, in which we showed that telomere dysfunction is actually connected to all hallmarks of aging. So think about it. You erode your telomeres, you activate p53, you increase p21, that causes senescence. The telomere dysfunction activates ATM, which phosphorylates YAP. YAP regulates pro-IL-18, which is a major cause of tissue inflammation, so-called inflammaging. Or I just mentioned mitochondria. Mitochondrial dysfunction, hallmark of aging. Well, through PGC, it's able to regulate that. And you could do this for almost any hallmark of aging. If you start to have one issue, it actually connects to all of the other hallmarks. And this is one of the reasons why regulating telomerase and regulating telomeres, we thought could be a very interesting way to attenuate the aging process or age-associated diseases.
Can you talk about, you had a nice study in Nature Aging in 2021, where you manipulated telomerase in a mouse model of Alzheimer's disease. And can you talk about that study and the findings there? I mean, you looked at not just what's going on at the cellular level, but you actually looked at clinically relevant endpoints in Alzheimer's, like cognitive function, learning and memory.
Yeah, so we were always interested in aging per se, as I mentioned, and we noticed that you have neurodegeneration and the telomerase knockout back in the 90s and so on and so forth. And so we're kind of always, this was always on our radar. But really what set the stage for the current experiments in the last few years was an observation where we generated an inducible telomerase knockout. So this is telomerase reverse transcriptase. And what we did in this experiment is we knocked in an estrogen receptor into the open reading frame of TERT. So we would generate a protein that was a fusion protein called TERT-ER. This now is regulatable by tamoxifen. And so in the absence of tamoxifen, TERT is denatured. It no longer functions. When you add tamoxifen, you can renature the protein, telomerase becomes active. So this allowed us to study the function of TERT under normal physiological regulation of TERT. And what we found was that when TERT was off, we got the same phenotypes that we saw in the knockout. However, when we turned telomerase back on, and these were animals that were very aged, they had diminished brains, microcephaly, they had impaired cognition, they were weak and frail, they had thin bones, alopecia, hair graying, and so on. They were really in very, very degenerative condition, about 1 month away from succumbing. In that state.
And this is in wild type mice?
That's correct. These
That's
are mice now that have, we brought them through successive generations,
right.
Now G3 or G4, so they're human-like telomere lengths.
Yeah.
The animal's prematurely aged.
Yeah.
And then in the prematurely aged mice, at the very end of their life, we turn telomerase back on. We were thinking, well, this might slow the aging process.
Right.
What we found was a reversal of aging. It's the first time that it was shown that aging is a reversible process, telling us that tissues retain a remarkable capacity to renew themselves if you remove the instigator of aging, in this case, telomerase and telomere dysfunction. However, what was really interesting, and in fact, somewhat annoying and inconvenient, which are always those interesting results, was that not only did proliferative tissues restore themselves, because when you have to go through S phase and cell proliferation in order for telomeres to be resynthesized. And we showed that they were. So think the intestine, skin, hematopoietic system. it would be somewhat expected that there would be some rejuvenation there. And we indeed saw that. And the germ cells as well were restored. However, we saw that the ejection fractions of the heart were improved. We saw that oligodendrocytes were now making more myelin. So the brains were shrunken, largely because of impaired myelin formation by oligodendrocytes, postmitotic. And so those postmitotic cells, hepatocytes, cardiomyocytes, neurons, oligodendrocytes, were being rejuvenated as well through a variety of different assays we did, both pathological as well as functional. So that was very interesting to us. And it then set us on a path to try to understand clearly what TERT was doing beyond its classical functions of synthesizing telomeres. And the other interesting thing that we found was that TERT is expressed in all somatic cells. Now that we have really good reagents to see TERT protein levels, TERT protein is in all cells, somatic cells. It's just lower. However, with the onset of aging, we found that TERT levels go down about 50% further. And we look at the promoters of the TERT locus, we see the accumulation of repressive epigenetic marks. And so we thought that perhaps that
methylation, DNA methylation?
Not DNA methylation, but more chromatin mark changes like H3K9, repressive mark, loss of H3K4 trimethylation, and so on. So the gain of repressive marks and the loss of activating marks in the chromatin. And so that was just an observation that was also in the back of our minds when we were thinking about all of this. So in humans in their 40s and 50s, depending on lifestyle, you'll see this repression of TERT just goes down 50%. And then in mice around 18 months of age, goes down about 50%. That's when mice begin to have onset of aging. Senescent cells accumulate, inflammation, immunosenescence, all those things, decreased neurogenesis, and so on. And so that was an interesting observation. But then the really key experiment was we looked at TERT het mice, young mice. So these were young mice that simply had 50% loss of telomerase reverse transcriptase. And we did transcriptomic analysis of the brain. And what we found were very interesting genes that were altered. BDNF, wow. HSP70, APP. So, maybe TERT is regulating transcriptional programs. And so what we then did was to do mass spec to show that TERT can interact with the mediator complex. And the mediator complex of transcription factors is essentially the bridge between a transcription factor that's sitting on a promoter that may not be all that active with the ability to bridge and connect to the enhancer of that gene. And so that what that then does is it increases the speed and processivity of the transcription factors. You get more mRNA of its target gene. And so that the implication of that was that interaction of TERT with the mediator complex may be influencing transcriptional complexes that are regulating gene expression programs, like BDNF, like HSP70, and so on. And all those genes are, as you know, are very important for Alzheimer's.
Yeah.
And so APP is amyloid precursor protein.
Yeah.
And we saw that it was increased with the TERT het. BDNF decreased with the TERT het. All very interesting. And so that was another piece of data. So that then motivated us to then look at Alzheimer's models. We both looked in mouse and in human model systems, IPS model systems.
Wow.
And we used the 3TG model and the FAD model for Alzheimer's.
I was involved I helped a little in generating the 3xTGAD mice because
that is a wonderful model.
Because we'd collaborated with George Martin, and he had a postdoc who'd made presenilin 1 mutant knock in mice. So presenilin is an enzyme that's involved in cleaving the amyloid precursor protein to liberate the amyloid beta peptide that forms the plaques. But those presenilin 1 mutant mice, they didn't have really any Alzheimer's phenotype. And it's because they had the mouse APP and the mouse A beta, which is a different amino acid sequence than human A beta. And the mouse A beta doesn't self-aggregate and accumulate. So then Frank LaFurla, who's at UC Irvine, well, I'm friends with him. He contacted me and asked if I could send him some of those mice. So what he did is he got male and female PS1 mutant knock-in mice, and he bred them, and he took out the one cell stage embryo and injected with expression vectors for mutant tau and mutant APP. So that's the 3 different genes. PS1 was a knock-in, and the other 2 are overexpression. And yeah, it's kind of slow, the process of accumulation of amyloid and tau pathology in their brain, but maybe that's good in some ways. It takes, like in our lab anyway, a year before you get like major accumulation, but it does happen, and I think it's a good model.
I think it's a good model as well, and thank you for your contribution. It's a very important one. So one of the things that we then did was to ask the question, well, in the mice, what happens with TERT expression early on in the development of pathology in these models? And as you mentioned in the 3TG model, a lot of things unfold in kind of the 6 to 18-month time frame. So we looked at 6 months, and we found that TERT was epigenetically repressed much sooner than what you see in a normal mouse brain, which takes about 18 months. So we saw that TERT was being modulated very early, prior to the onset of pathologies. So then what we did was did a transgenic experiment. We always like genetic ground truth first. So we did a transgenic experiment in which we maintained with a CAM promoter, maintained normal levels of TERT in neurons. And that allowed us to then ask, well, if we maintain TERT in the 2 models, what happens? We also did IPS human neurons from demented and non-demented controls. And this is the duplication of APP model system. And what we found was that if we constitutively maintain TERT in those IPS neurons, that we can block the accumulation of A beta that occurs in those models. And then we also looked at gene expression, genes that were involved, BDNF, HSP70, synaptic proteins, virtually all genes involved in learning and memory were affected. Antioxidant genes were affected as well. And so that was the human data. But in the mouse, when we constitutively expressed the transgene, we were able to block many of the phenotypic hallmarks that you have described in the past, which was the accumulation of intracellular amyloid and extracellular plaque formation, number 1. At the EM level, we could see that the dendritic spine density was maintained. We could see that at the transcriptomic level, hippocampal transcriptomes, that virtually the number 1 genes that were altered were learning and memory genes. And then we can show that cognition was preserved using Barnes maze and so on. And so that told us that TERT plays an important pathogenetic role in the development of the phenotypic hallmarks. And that suggested that perhaps maintenance of TERT might be a novel therapeutic strategy and so on. And then finally, we did some additional biochemical studies to ask, well, in neurons, what does TERT actually interact with. So I mentioned it interacts with transcription factor complexes. Well, it turns out it interacts with beta-catenin. And
Yeah.
as you know, beta-catenin is the master regulator for learning and memory genes. Regulates many, many genes involved in synaptic biology and all the things that are relevant to learning and memory. And so we've that kind of closed the loop to a certain extent with that genetic experiment.
And
Having that,
also, it's you had evidence that the telomerase protein is also regulating the Krebs,
correct, yeah,
which is another very important transcriptional regulator of learning and memory related
correct,
genes. Yeah.
exactly. And we haven't really quite dissected the biochemistry of that and what's important and what's not, what interaction, how does it interact with the mediator complex and so on. So this is still descriptive, but the downstream consequences are quite clear, and that is that you clearly have that. And the other thing that we did in the human model system is that when you knock out or you generate a TERT inactive molecule and constitutively express that, so catalytically inactive, can't synthesize telomeres, that is able to still regulate gene expression.
Yeah.
And so again, that kind of the nail in the coffin that they're clearly non-canonical functions of TERT. And then what was really a key next series of experiments began a little bit earlier than that. And that is we wanted to discover pharmacologic agents that could de-repress TERT locus, that could restore its normal youthful levels, in the hope that that might attenuate normal aging or age-associated diseases like Alzheimer's. Now, to do that, we embarked on a very particular way of trying to discover such compounds. So first, we took the human back transgene, which had 160 kilobases of the human TERT locus. And we wanted to see if the cis elements that regulated TERT in a human were all present. And so what we did is we put that into a mouse, and we showed that that recapitulated the temporal and spatial expression of human TERT in the mouse cells. So we now knew that the cis elements were all there. And in fact, the regulatory components that in the mouse that regulate that is also there. And so we felt that the mouse with this back transgenic, which we knocked in a reporter, might provide a good platform to screen close to 1 million compounds. And we did that in collaboration with Pete Schultz at the Scripps. And we then identified a couple of compounds, one of which was reported in the Cell paper. And what we found was that when we took that compound and put it onto normal human cells or Werner's fibroblasts, progeroid syndrome fibroblasts, we can.
So I was a lab chief at NIA for 20 years before I retired, and another lab chief there is Will Bohr. And so you mentioned DNA damage, response, and repair, and these progeroid syndromes that Ron's talking about are caused by mutations in DNA repair, defective DNA repair. So Werner, there's cocaine, and there's some others. Okay.
The blooms and so on, yes.
Yeah.
And in fact, in the mouse, if you knock out Wern, you do not get a phenotype. And we showed 20 years ago that if you now make telomeres in the mouse human-like and more limiting, you then get little Werner's patients. You recapitulate the phenotype. So telomeres play an important rate-limiting pathogenic role in the development of these progeroid phenotypes. And so it's, again, yet another example of how important telomeres are in pathologic aging in this particular case. And so what we then did was to take this compound, which we call TERT activating compound, or TAC, and we applied it to human cells. And indeed, we can find that these human cells can now increase TERT expression by about 50 to 100%, de-repress the TERT locus, extend telomeres, extend replicate potential, and also eliminate the DNA damage foci signals at telomeres. So classical functions of telomerase. And then what we did is we administered the molecule to mice. We did this IP, but it is orally bioavailable as well, but we did it through IP injection. And we just treated the mice with 3 times per week. And these were mice that were normally aged mice. So we started when these mice were 20 to 22 months of age. This would be equivalent of somebody in their 70s. We treated them for 6 months, equivalent to somebody in their 90s. Mice typically live around 30 plus months in general, 30 to 36 months, depending on how you keep them, depending on the strain and so on. And so very end of, last quarter of life type studies where we have a lot of aging symptoms at 22 months of age. The tissues are chock full of senescent cells. You have tissue inflammation, lots of TNF alpha and IL-6 and interleukins 1 alpha and beta, so on and so forth. And impaired cognition. You look at the hippocampus, there's no neurogenesis, and so on. And so what we then did was to analyze the mice at many levels after 6 months of treatment. So on the pathologic level, we found that we eliminated senescent cells, and we understand the mechanism for that, which we can get into. We eliminated or dramatically reduced inflammaging, so circulating cytokines went down, and also by IHC, inflammatory cytokines were also greatly diminished in tissues, kidney, brain, and so on. And then we also looked at a number of specific tissues. In particular, we focused on the brain since that's such a critical organ system for aging. And we found that microglial activation was dramatically decreased. So for the aficionados, we looked at IBA1 marker, but we also looked at soma size, and they were dramatically reduced and attenuated in their activation state. So that was down. We also looked at inflammatory cytokines. So TNF alpha, which is a key regulator of many aberrant cytokines, was also dramatically reduced by multiple fold.
Whoops, Ron?
And then we have the cortex, and found that we maintained nice arborization and maintenance of the brain. And then we did some cognitive studies and found that in Barnes maze, this is the escape hole, learning where the escape hole is, novel object recognition, or the Y maze test, the propensity of mice to explore unknown territory versus not, that all reflections of learning and memory, that all of those were dramatically improved. Then we also looked in the muscle and found that key master regulators of muscle, of myogenesis, MyoD, myogenin, PAX3, for example, were upregulated. And we found that that was correlated with enhanced grip strength and enhanced coordination and balance on the rotor rod test.
Yeah.
And so there are many things that we're still looking at. And so those are all things that told us that the maintenance of TERT provides a potential therapeutic strategy for attenuating hallmarks of aging at multiple levels: senescence, inflammation, proliferative capacity of cells, think immune cells, cognition, and so on. And then finally, we did unbiased analyses of the hippocampus, because what we saw there was neurogenesis in the hippocampus in 28-month-old mice, which you don't see. It's completely devoid of new neurons. And we showed that there was a new neuron formation, and we also used markers that showed that those neurons matured into neurons, and that correlated with, of course, the enhanced cognition that we saw in those mice as well, because neurogenesis in the hippocampus is important for the maintenance of working memory.
Yeah.
So when you get chemo brain, for example, you wipe out those neurons, and you get your memory is impaired for a good 6 months until they start coming back after cessation of treatment and so on. And so when we did transcriptomic analysis of the hippocampus under TAC treatment, we saw signatures of stem cell, of dopaminergic neurogenesis, synaptic proteins, and telomere maintenance, of course, because telomerase was up. And then the only category that was down was inflammation, cytokines, and so on. So this gave us a really clear perspective that TERT modulates key pathways that are integral to normal, healthy aging in the brain.
And with TAC or the other chemicals you've identified that can restore telomerase levels, what's your plan now? Or you've maybe already done this. Have you administered TAC to different mouse models of age-related disease, or are you going to leap to safety studies in humans with some particular kind of, what's your vision for the future of both basic and translational research? Because there's, what you're describing is quite remarkable. It's exciting, and it's really a fantastic series of studies you've described, elegant studies, the genetic manipulations in the mice, and then, you know, that we can't really do those genetic manipulations very well in humans yet. So that's not a practical thing, at least now. I don't know, who knows with CRISPR and so on in the future. But so what's your moving forward? What are you going to do? Because it's really exciting, and you have to kind of pick and choose. For example, if you go to human clinical trials, are you going to do a disease? Are you going to just do old people and make them young? Or what do you...
Well, again, there's a lot to unpack there, but
Yeah.
so first, we will continue to conduct our science and test TAC in a variety of different systems, such as the Alzheimer's mice that we talked earlier. Can we recapitulate what we saw genetically in the context of the mouse? And so those studies are underway, and our preliminary data suggests, and we haven't done cognition, but our preliminary data suggests that it modulates amyloid and neuroinflammation. And so we're hopeful that we may see similar kinds of findings and so on. And there are...
is that still with intraperitoneal injections, or can you put it in food? Can you put it in food? Or
yeah, so we haven't done the formulation. Yes, you could put it in food, etc. Those are all things that we will do. And those are important as well, because you don't want to subject mice to IP injections if you don't have to. To stress in and of itself. And so we are very interested in doing it a variety of different ways. And so there is the science is continuing, and we're testing it in a variety of different systems, so on and so forth. In addition, we are motivated to move this forward into its application for human biology. And I want to emphasize here, the anti-aging field has been marred by, let's say, irrational exuberance. And so I really want to emphasize that while the mouse is a great model system, there are more surprises than fulfilled prophecies when it comes to clinical application.
Yeah.
And so it'll be very important for us to see if what we've discovered in the mouse is applicable to the human situation,
Yeah.
or even aging dogs or primate Alzheimer's models and other things that would put us in a good position to give us confidence that we should really drive this very hard and very deliberately into a variety of different applications on the human side. Think chemobrain as an example. The number one cause of dose continuation of 700,000 people that go on certain chemotherapeutic agents that cause chemotherapy-induced peripheral neuropathy or chemobrain is that side effect. And so can we attenuate TERT?
Yeah.
We see new neuron formation as a major problem for prolonged space travel. And NASA is interested in asking whether or not this modality could potentially better enable interplanetary travel to maintain cognition. Because as you know, when you have all these alpha particles going through your brain, it's not very good for your brain over the course of time and so on. So there are many, many different applications. Sarcopenia is a major cause of morbidity in elderly individuals. That it's a very significant fraction. It's a $40 billion hit on healthcare, et cetera. So the idea here would be to look at once you get past safety studies and formulation, et cetera, and you would do first in human studies, and those studies, even the normal human volunteers and an aged individual, would be able to have some data-rich analyses that would tell you whether or not you're reducing circulating cytokines, increasing telomere lengths into peripheral blood lymphocytes, things of that nature, senescent cells, et
The
cetera.
sarcopenia is a really good target, it would seem, because it's, I mean, if you described effects on the muscle, right? And increased grip strength.
Well, it's also the neuromuscular, right? It's
Yeah,
neuromuscular improvement. So as you know, the neurons play a
sure.
super important role. It's not just what happens intrinsically in the satellite cells, right?
Yeah.
So you sort of need both, and we think that we may be able to enhance both ends of the equation, although we haven't specifically by EM looked at synaptic vesicles in the neuromuscular junction.
Have
That
you
in the
come across Luigi Ferrucci in your times? He's the scientific director at NIA, the Intramural Research Program. He's got a lot of focus on sarcopenia, but
Good.
yeah.
Yeah. So yes, we're very interested in working with the field, working with NIA. I've always been involved in large, collaborative efforts because the collective wisdom of groups and capabilities is always stronger, and people are dying, and
Yeah.
you have to have a sense of urgency to really make an impact on pain and suffering from a number of different conditions. Obviously, since aging itself underlies age-associated diseases, if this does have an impact on normal aging processes, there's potential that it could attenuate, age-associated disorders as well. But those are all studies that really have to be proven rigorously in safety studies and in efficacy studies. And this would put us in a position to then do, a series of initial studies like sarcopenia, for example, or chemobrain. And then would do more extensive studies such as Alzheimer's, Parkinson's would be another, obviously, with the twofold increase in presynaptic vesicles. You would think, and dopaminergic neurogenesis, you would think that might be a fertile ground for opportunity to help people. And then you ultimately have aging itself, and that's a disease that has 100% incidence and 100% mortality. But the FDA has yet to do an aging study. We don't know what that kind of study would look like. We don't have surrogate markers as we do with statins and cholesterol or blood antihypertensives and blood pressure. We don't have those surrogates. In those studies, we don't look for strokes and heart attacks as endpoints, right? It's safety and it's affecting those surrogates. Whereas in aging, we don't yet have agreement on what those surrogates would be. Would it be cellular methylation patterns, chromatin marks, a senescent cell accumulation, inflammaging, that if you were to interdict those, would you then say this might be something that would be useful for individuals that are walking the telomere plank like we are?
So you mentioned grip strength, right? I actually have one. I don't know if you've got one of these, but this is a thing you can test your grip. But grip strength and walking speed are good predictors of longevity. So it seems like, if you take an healthy aged individuals, and first, yeah, obviously do safety studies and so on, and then I don't know how long you go, maybe a year treatment or something, and then just look at,
It's actually 6 to 12 months. There are endpoints at 12 months, and there are very, very rigorous parameters for sarcopenia clinical trials.
Yeah. But I'm thinking in people without sarcopenia, because
Yes.
yeah.
No, but I think that frankly, without sarcopenia, when you get to be over age 80, the a very significant fraction of those individuals are going to have significant weakness of muscles. And that puts you in a position to get a lot of have data-rich normal human volunteer studies where you'd be able to ask, you could look at, actually even simple things like glomerular filtration, just from your chemistry, blood chemistry. You could electronically know whether or not your podocytes are working better. You could look at your cardiac ejection fraction. You could,
or even heart rate variability.
You could look correct. You could look at do some simple cognitive tests.
Yeah.
So there are many things, and then you can look at certain molecular parameters, and those are all things that we would likely do as we bring this forward into clinical testing.
Okay, Ron. So in the meantime, listeners and viewers, exercise, moderation in calorie intake, keep your neural circuits in your brain intellectually engaged. We know those things are good. So, don't count on TAC making you young, but it's very exciting. And, based on what I've seen of your data, looking through your papers, this has a lot of potential. So anyway, this is, I agree 100% with you, Ron, that if we understand and be able to manipulate aging processes, we'll have a big impact on a lot of different diseases, which most of the things that kill us are age-related.
Yeah, I think the GLP-1 agonists are a good paradigm for this, starting out in diabetes. Oh, you lose weight. Oh, you improve cardiovascular function. Oh, you actually have less cancer. So,
but it's inhibiting appetite, so it's actually causing calorie restriction, and probably that is a big component in my view.
Unfortunately, it also reduces muscle mass dramatically, because evolutionarily, when you restrict calories, this is why exercising while you diet is very important, because when you restrict your calories, the first thing your body does is it tries to reduce your muscle mass, which is the major consumer of energy. And so you see individuals that are, prisoners of war and or individuals that are undergoing famine. They're essentially bones. They have very little muscle mass. And so that, is an indication that, we still have a ways to go so that we can develop strategies that really go after the fundamentals, right? So having GLP-1, I think, is a major breakthrough. There's no question. It is one of the great medicines of all time. But from my perspective, thinking back to how do we maintain cell state? Why do cells change? at this transition of midlife from young to old. And if we can understand the normal physiological processes that are co-opted, then what we can do is to leverage that knowledge in a way that allows us to then reset cell state. And folks are trying to think about this with the Yamanaka factors and so on, and reprogramming epigenetically. I think there is even greater challenge because of the incidence of teratomas and things like that, because it's a nuclear bomb.
Yeah.
When you're starting to mess around with, with those kinds of transcription factors. But the idea would be here that you would essentially take advantage of something that's evolutionarily conserved, which is TERT's ability to regulate in a subtle way gene expression programs. You're not talking about major changes like the levels you see of TERT in a cancer cell, which are tenfold higher. What you're doing is simply restoring physiological expression. So if there are strategies like that, or even other strategies that others will discover, that will allow us, put us in a position to really reset cell state. And that could potentially enhance the lifespan of individuals to diminish age-associated diseases.
Yeah. Well, I really appreciate you taking the time, Ron. I'm retired now, and the mornings I spend doing this and writing, and the rest of the day goof off. But your time's very valuable. I appreciate you taking the time. It was nice to meet you. I've actually followed your work for decades, even though we didn't really do much work in the exact same area. And
Thank you.
I'm glad you're have a lot of emphasis on the brain now.
Thank you very much. And the work that we've done over the years is really a testament to the exceptional trainees that I've had and collaborators that I've had over the decades. So very, very fortunate to have such a gifted group of people that have taught me more than I've taught them.
It's ditto for me over my career. Okay, thanks, Ron.
Thank you. Have a nice day.
You too.